Hello everyone, I am Shweta, doing first MSc Microbiology. We are going to discuss about Hepatitis B virus. Introduction. The term hepatitis describes inflammation of the liver. The hepatitis may be caused by drugs, alcohols, autoimmune diseases, metabolic diseases, and viruses. The viral infections persist for more than half the cases of acute hepatitis. There are different types of hepatitis virus are hepatitis A, B, C, D, E, F and G. The hepatitis F is not separate entity. It is the mutant of hepatitis B virus. All of these are RNA viruses except the hepatitis B virus is a DNA virus. The viral hepatitis is classified as acute hepatitis and the chronic hepatitis. Hepatitis B virus. Hepatitis B is caused by the hepatitis B virus. It is an enveloped virus containing a partially double-stranded circular DNA genome and it is classified within the family of hepadnavirus. The virus interferes with the functions of liver while replicating in the hepatocytes. The immune system is then activated to produce a specific reaction to combat and possibly to eradicate the infectious agents. The hepatitis B virus may be the cause of up to 80% of all cases of hepatocellular carcinoma in the worldwide. Structure. Hepatitis B virus is a DNA virus with a remarkably compact genomic structure. It has a circular partially double-stranded DNA virus. It is small, circular, 3200 base pair per size. Hepatitis B virus DNA codes for 4 sets of viral products and it has a complex and multi-particle structure. The hepatitis B virus is 42 nanometer in diameter and composed of 27 nanometer nucleocapsid core (HBC) antigen surrounded by outer lipoprotein coat, also called as envelope, which is containing the surface antigen HBs antigen. The core antigens are located in the nucleocapsid. This is the picture of hepatitis B virus. Pathogenesis. The hepatitis B virus is transmitted by parenteral or mucosal exposure to HBs antigen positive body fluids from persons who have acute or chronic hepatitis B virus infection. It replicates in hepatocytes through a unique reverse transcription process. It has three steps: entry, multiplication, and spread, and the last, liver cell damage. The first one is entry. The virus gains entry by the following routes: through blood transfusion, sexual transmission, from infected mothers to neonates, through contaminated syringes and needles. In rare cases, it is transmitted by arthropods. Multiplication and spread. The target of hepatitis B virus is the hepatocyte. HBV proteins and genomes were identified in extrahepatic sites, also bone marrow, spleen, lymph nodes, and circulating lymph nodes, but they do not produce any damage in these locations. The steps involved in the viral multiplication and spread are: the first, virus attaches to the hepatocyte with the help of its surface antigen. HBV enters inside the cytoplasm through receptor-mediated endocytosis. The DNA of the virion is transported into the nucleus. DNA transcription follows, resulting in the formation of mRNA. The short mRNA transcripts undergo translation in the endoplasmic reticulum. The translation products are pre-spike and spike proteins. P-protein translation occurs in the cytoplasm and it is coupled to capsid protein formation. P and capsid proteins are assembled and they enclose the DNA. Replicated DNA undergoes nick formation resulting in double-stranded DNA with a staggered end. staggered end star. Morphological changes occur on the surface of plasma membrane, and the last, complete virions are released through the process of exocytosis. The third one is liver cell damage. Replication results in injury of hepatocytes and release of progeny virions into the bloodstream. Cell injury is not only caused by cytophatic efforts of virus but also by the activation of cytotoxic immune mechanisms. This results in liver tissue degeneration and the release of liver associated enzymes into the bloodstream. This is followed by jaundice, the accumulation of bilirubin in the skin and other tissues with a resulting of yellow appearance. The chronic hepatitis leads to persistent hepatitis with mild periportal inflammation. This will lead to chronic active hepatitis with more widespread inflammation and necrosis. The chronic active hepatitis leads to cirrhosis and hepatocellular carcinoma. The symptoms of hepatitis B virus include fever, loss of appetite, nausea and vomiting, abdominal pain, weakness and fatigue, joint pain, jaundice, which is yellowing of skin and whites of eyes, dark colored urine, and swelling with fluid in belly or arms and legs. Laboratory diagnosis of hepatitis B virus. Several types of blood tests are recommended for diagnosis of hepatitis. Liver enzyme test, reduction of hepatitis B antigens. In liver enzyme test, blood levels of liver enzymes such as alanine aminotransferase and aspartate aminotransferase are elevated in the early stages of viral hepatitis. In the detection of hepatitis B antigens, ELISA test is used for the detection of HB surface antigen. The ELISA can also be used for the detection of hepatitis antibodies. Treatment. People with chronic and active inflammation of liver cells due to hepatitis B virus are treated with interferon. It blocks the entry of virus into the cells. In recent years, interferons is prescribed in combinations with ribavirin. Interferon is not recommended for people who have developed cirrhosis or scarring of liver tissue. For those, lamovidin is recommended. Medicine decreases the multiplication of viruses. Interferon stimulates the body's natural defenses. During interferon treatment, the following side effects are observed, such as discomfort, body aches, fatigue, irritability, loss of appetite, nausea, and the diarrhea. To cope with the side effects of interferon, the following measures are followed: intake of large volumes of water every day, intake of painkillers with the help of recommendations by physician, resting as much as possible, practicing relaxation techniques such as breathing exercise, regular food habits, and frequent brushing. In case of severe nausea, medicines are taken as recommended by physicians. To prevent the hepatitis B virus, prevention is possible by active and passive immunization. Vaccine injection is given in muscles of the upper and outer parts of the arm at birth, at the age of 1 month and at 6 months. A booster dose is recommended at 5 years of age. Two types of vaccines are currently available for the hepatitis B virus. Recombinant HB vaccine synthesized from yeast cells are safe and effective, and it provides 90% protection. And the second one is plasma derived vaccine. Thank you for your patience.